Drug discovery today takes over a decade and can cost billions. At Ainnocence, we're changing that by using AI to make drug development up to 80% faster and more affordable.
Hi, I'm Dr. Lurong Pan, founder and CEO of Ainnocence. We are combining machine learning and AI and molecular sciences to speed up the discovery of new medicines and help bring better treatment to patients faster.
Finding a new drug is like looking for a needle in a molecular haystack. Researchers test thousands of compounds by hand, most fail. The process is slow, expensive, and inefficient.
Our platform uses AI to predict how a molecule would interact with the target before any step into the wet lab. We eliminate weak candidates earlier, saving time, money, and resources.
Faster discovery means faster delivery. By reducing the early-stage timeline, Ainnocence helps get effective treatments to patients who need them without years of delay.
When we cut through inefficiency, we give scientists a head start and give patients a chance at better, quicker outcomes. We've seen our AI identify promising drug leads in weeks, not months. It can scan vast compound libraries, antibody libraries, evaluate molecular fits, and even generates novel candidates we won't have found manually.
Ainnocence is expanding, working with biotech partners, researchers, and pharma innovators to scale AI-driven discovery around the world.
We're excited to keep building, collaborating, and pushing the boundary of what AI can do in life science.
If you're excited about the future of AI and drug discovery, we want to hear from you. Leave a comment, ask a question, or visit www.ainnocence.com to explore how Ainnocence is changing the game and how you can get involved.
Let me introduce SentinusAI, the protein design engine from Ainnocence. We have three major platforms that facilitate the drug discovery process. Sentinus is the protein design engine, Carbon AI is the small molecule design engine, and Cellular AI, the cell programming AI engine. Today, I'm going to present the SentinusAI core functions. So first of all, in using this platform, we no longer need animal immunization, which is an ethical technology without animal involvement. And then we have fully optimized the engine to first save about 10,000 times of computational cost, and at the same time being tested over 60 projects with good success rate up to 90%. And the entire technology platform is able to design antibodies against even target that without any 3D structure, which expanded the therapeutic targets by over 70%, that most of the target cannot be computed in the conventional CADD technologies able to compute using our platform. There's a simple comparison between our platform and the traditional antibody screening technology, such as hybridoma, phage display, and animal immunization. All that comes with a shorter time and a lower cost and a much better success rate, and without animal study. And we have multiple AI models to optimize different aspects of a antibody or protein drug, from efficacy, mostly optimized affinity against a therapeutic target, and at the same time avoid any unwanted off targets. And we can optimize the safety, evaluate the immunogenicity, and perform the in silico humanization. And then we have the developmentability, which are able to optimize the concerns during manufacture. And the entire technology workflow works very simple. There's only take two major steps. First, we take the input of an interested sequence of the therapeutic target, and then we put the information in the AI engine, and only take about a week of computing. We can screening billions of candidates and recommend about 10 to 100 candidate molecules to perform wet lab test. The first process only take 1 week for computation and about 2 weeks for wet lab validation. And we normally would do another 2 or 3 rounds of optimization based on wet lab data feedback. We call it reinforcement learning until we get the best candidate. Here's some case studies of what this engine can do. First, the typical case is affinity maturation. So we take a reference antibody with certain affinity to a target, but not ideal. And then we use AI to compute actual molecules that are similar to this target, but predicted to have a higher affinity. And as you can see, normally in the first round, we will be identify a few more hits with better affinity. And then we do the second round that the affinity further increase, and also the positive hits, or number of positive hits also increase. And this process only take a couple of rounds that we could increase about 10 to 100-fold in terms of affinity to a specific target. And we have many clients that bring us either a failed project or a competitor's patents with the reference antibody to start with, and we'll be able to quickly make a successful project for them within a month or two. Another case is a de novo antibody design. So in this case, we won't have any starting point of reference antibody, but we will have just the sequence of target of interests. In this case, where first round will generate dozens of very diverse antibody sequence in different format. We'll be able to generate from VHH, Fab, or a different variety of monoclonal antibody or multispecific antibody, the form as you designed. And then we will perform our computation. Normally the first round we will have some weak hits, and then we further do another round of optimization, we will have better hits. The third function is the epitope mapping. For some novel target or even some known target that people would like to know which specific epitope are related to its actual in vitro or in vivo function. So in this case, we'll be able to scanning all the possible epitope along with the therapeutic target and quickly generate a score that rank the importance of the binding. And the next one is the developmentability. So using the DivaPro score system, we can score different aspects of the target from unwanted motif, isoelectric score, thermal stability score, and then integrate all the score into one scoring system so that we can quickly enumerate possible change on the sequence to improve the overall developmentability of the product. Another common request is a humanization, because in the old way, a lot of antibody are discovered through animal immunization, which we would take the animal's form of antibody and to start with and has to humanize it before it enter clinical trial for human. In this process, we have trained a deep learning model be accurately differentiate between human and animal species, and it will be able to generate only human sequences and at the same time modify existing animal sequences into human. And this process is also done by AI automatically with high precision. Another useful function is off-target screening. In this function, we will be able to screen all the membrane and circular proteins just by the panel that you want. We can do all that or just some of that for your therapeutic discovery. And in the past, it's very costly to purify all the potential off-target and do a wet lab screening. But using our engine, you can do that just with a click away. So our project life cycles, a typical fee-for-service module, start with signing the NDA, and then we will exchange some information of the project, and then we provide the quote and the statement of work. And after we execute the contract, a down payment will be requested, and after that we'll start the project. And after we have expressed and tested the antibody and to make sure it's satisfied with the initial standard, and then this project will be delivered, and then the final payment will be paid. So that's a very simple project process. We have the fee-for-service model, and we also have the co-development model that the second one will involve more deeply in the entire life cycle of the pipeline and do unlimited optimization whenever needed in the process. And we will claim some royalty of the pipeline as well and make sure its final success. So if you're interested in our service, here is our contact information for career service and all other inquiries.
Thanks for watching. If you found this helpful, give us a like, subscribe for more, and share it with someone who cares about the future of healthcare. This is just the beginning.